Hi, my name is Melo. I'm co-founder and CEO of Menten AI. Love to walk you through our work today. Let me begin with a fact. Up to 80% of drug targets are beyond the reach of small molecules or biologics. Now, when you think about the fact that 90% of drugs today are in fact small molecules and biologics, you see that there's a huge disconnect between our unmet medical need and the methods, the drugs that we have available today. Now, at Menten AI, we believe that we're in the middle of a paradigm shift where new technologies, and in particular AI, will help us accelerate this drug discovery process. In particular, we've been focused on achieving three things. First, understand drug targets better. Second, design drug modalities beyond small molecules and biologics. And third, to accelerate this whole drug discovery process. We've been focused on cyclic peptides as a therapeutic modality for a number of reasons. They have advantages between small molecules, advantages over small molecules and biologics. And in recent months, they've seen a lot of interest, in part due to success in the clinic, as exemplified by Merck's PCSK9 oral cyclic peptide inhibitor and J&J's IL-23 cyclic peptide inhibitor as well. And we believe in the coming decade, cyclic peptides will have a growing role in the industry. Now, at Menten AI, we focus on developing an artificial intelligence platform that allow us to create these cyclic peptides at scale, targeting new chemistry. Now, in particular, we focus on developing a system that allows us to create cyclic peptides with the right drug properties from the very beginning. In order to do this, we develop multi-parameter design where we're able to take into account not just potency, but also solubility, stability, permeability, and other properties, and design with those simultaneously. I would love to walk you through the platform. We begin by taking a target protein of interest and applying our proprietary machine learning and molecular dynamics models to help us understand the target better and identify new druggable surface interfaces for design. As an example of this, you see here TNF-alpha, one of the most well-known proteins in drug discovery, and in fact, the drug target of the drug Humira, one of the best-selling drugs of all time. And when we ran this through the platform was able to identify a novel binding pocket that was previously not utilized by any available drugs. And when we actually used this pocket in design, we saw that many of our most potent hits in the nanomolar range were in fact applying using this binding site. Now, once we have a input target model, this is the core of the platform. We apply our molecular engine to design completely novel cyclic peptides from scratch. The platform works by designing a backbone, then bringing in all kinds of natural amino acids to design the sequence of the structure. And finally, we apply machine learning to help us rank the designs in a very systematic fashion. The output of this is truly drug-like peptides. And what do I mean by drug-like peptides? We've seen potencies systematically in the nanomolar range, and we've tested this across dozens of projects, hundreds of peptides synthesized and experimental results. We've seen over 80% hit success rate. We see plasma stability in the order of hours. We now see permeability, which is important, will allow us to take these drugs orally and make them cell penetrant. And we believe a lot of this success in drug properties is driven by the chemical diversity that we're able to build into these cyclic peptides. We, in fact, support over 1,300 amino acids, and this includes L, D, amino acids, as well as a variety of chemical elements as well. Now, once we have a hit, we're actually able to optimize it further. And we built a optimization engine that allows us to take hits, whether discovered through an in silico platform like ours, or through some experimental display technology, such as phage display, mRNA display. And we are then able to take these initial hits and drive potency or whatever other property is desired using our proprietary machine learning optimization engine. As a succinct example of this, we took Menten 048, one of our hit molecules in one of our initial programs, which showed our good activity, potency, and solubility, but did not have enough exposure for in vivo efficacy. So we perform a design cycle to optimize this molecule, which then from which we derive Menten 296. Which saw optimization across the board in potency, log improvement in exposure over time, which was then finally strong enough to get us that in vivo efficacy in a mouse IBD model. Now, bringing all of this technology together, we're able to go from target selection, generative AI generation, and in vitro efficacy in a matter of months. In fact, we've done this in 6 months, and most importantly, with fewer than 100 molecules. This contrasts strongly with many years of research and development and testing thousands of molecules, which is routinely done in the industry today. By bringing these technologies together, we're able to understand the drug targets better. We're able to design molecules beyond the reach of small molecules and biologics, and we're able to accelerate the entire process. We've been focusing on partnering with top pharma, and we have a number of areas of interest, in particular, development of oral and cell-permeable peptides, as well as bispecifics and optimization as well. As I said, we've been working with top 10 pharma, and we're open to collaboration with others as well. If you have any questions or would like to learn more, feel free to reach out to me. Myself and the team will love to learn more about your program. Thank you so much.